Hi, this is Sarah from the Highlands Center for Natural History. Today I'm at Granite Creek at the Watson Woods Riparian area, just pondering life. Specifically, I'm pondering the life that's unseen to the unaided eye. If we want to better understand our freshwater ecosystems here in the Central Highlands and beyond, then we need to understand microorganisms. Tiny single or multicellular life forms living all around us, but especially in and around water. Examples of microorganisms include bacteria, fungi, and algae. Filamentous algae, like this, is one of the reasons I got obsessed with creek exploration. It's slimy, and it pulls apart in strands or filaments. It's really fun to play with. And it's a perfect example of what we call a biofilm. This is a collection of microorganisms clumped together. Sometimes microorganisms create substances to stick together and form colonies or communities. And these biofilms are wonderful examples of the microbiological world around us. So here's what we're gonna do. I'm gonna share with you one of my favorite experiments for growing biofilms and for learning more about soil biology, microbiology, and freshwater ecology, the Winogradsky Column. Winogradsky columns are closed systems of soil and water, which over months and years develop biofilms of different colors that'll teach us about the microorganisms living in our soils and water. Let's head down to the pond and make one. For this experiment, you're gonna need a sealable container. In this case, a 2-liter bottle. We need it to be sealable because we don't want anything to be able to get in or out. We want to create a closed system where everything cycles within the bottle. You're also going to need a mud and soil sample. So this is the funnest part. I'm going to move away some of this top layer, including a lot of the leaves. And then I'm going to start to scoop out some of this really thick mud and put it in my container here. So now that we have our mud sample, we're gonna add some things to it to jumpstart the growth of microorganisms. The first thing we'll add is some sort of cellulose or carbon. This is newspaper shreddings, but you can use dry leaves or anything that you know has a lot of carbon in it. Next, we need to add some sort of sulfur. Eggs and egg shells have a lot of sulfur in them, so this is a mixed up egg and I put the eggshells in it. Now this is optional, but you can add some sort of multivitamin crushed up because that's going to have trace amounts of things like magnesium and calcium, which could add some interesting growing to your column. Once we have our concoction, we can start to fill our bottle. And this can take some time, so be patient with yourself. You can add water to make it a little more liquidy, or add something that has a little more sand in it, and that'll help it flow a little better too. Remember, wash your hands when you're done, or wear gloves. Next, we're gonna top off the whole thing with a little bit of water from the same location that you're taking your sample. We don't want to use tap water at home because it might have chlorine or other things that could affect our experiment. The water is going to do two things for our system. The first thing is it's going to keep things alive. Just like the algae is going to dry out in the sun and die, the same is true with other microorganisms. Without water, they aren't able to live. The other thing this water is going to do is it's going to create two circumstances in our system. One is going to be an aerobic environment where there's a bunch of oxygen, and the other is going to be an anaerobic environment down below where there's no oxygen. So now that your column's complete, you'll want to label it with the date and the location of sample. Then when you get home, you're going to look for a spot to put your column. It needs to have three things. One, it needs some sunlight. A lot of the microorganisms living in here need sunlight in order to survive. Two, you don't want it to get too hot. And three, you want it to be in a spot that's going to be undisturbed. This is a long-term experiment. So that means you might not see any biofilms develop for months or even years. However, the long-term nature of this experiment is one of my favorite parts. I've had Winogradsky columns for years of my life. There's some universities that have been growing them for decades. The excitement of seeing the reds or purples or blacks develop and this evidence of these microorganism communities is super exciting. So I will check back with you as mine develops, and I hope you'll do the same. Thanks for joining me for this Winogradsky fresh air moment. I'm Sarah from the Highlands Center. I'll see you again soon.